Hi, my name is Yury Bukhman. I'm a computational biologist at Morgridge Institute for Research in Madison, Wisconsin, USA. I'll be talking about the annotation of cell types in thymus single-cell RNA-seq datasets. This work is part of a project aimed at growing thymus organoids and xenografts from induced pluripotent stem cells. The role of the single-cell RNA-seq experiments is to assess if the organoids contain the cell types that normally occur in the thymus. We are evaluating two alternative and potentially complementary approaches to cell type annotation. One is reference-based, utilizing an extensive annotated thymus cell atlas and a deep learning model trained by a selects gene known as scVI. The other is reference-free, utilizing ChISA, an agentic LLM-based tool developed by the Kinziorski lab at UW-Madison. In the reference-based workflow, we use the scVI model to project both query and reference datasets into the same embedding. We then train a random forest classifier on the reference and apply it to the query to predict cell types. We leverage the cell ontology to annotate cell types in a hierarchical fashion. We first partition the dataset into high-level cell types, then find subtypes of each high-level type. In order to test our methods, we pick two random representative subsets of the thymus cell atlas and use one of them as the query and the other as the reference. After running the annotation workflow on the query dataset, we compare predicted cell types to the original cell atlas annotations, treating the latter as the gold standard. Since in this exercise both the query and the reference are drawn from the same cell atlas, there is no batch effect, as you can see on the UMAP plot on the right. The heat map on the left shows that there is a nearly perfect correspondence between predicted cell types and the original selects gene annotations. The violin plot on the right shows that the scores of the correct predictions tend to be higher than those of incorrect ones. When we project the data from different studies into an scVI embedding, some batch effect remains. The UMAP on the left shows a patient sample and the Yayon thymus cell atlas. Although the clustering of cells is driven primarily by major cell types, some batch effect remains as well. The Yayon atlas itself is composed of 5 studies. The plot on the right shows that there is a batch effect between those studies as well. Note, however, that thymus preparations usually include thymocyte depletion procedures aimed at boosting the fraction of stromal cells. Different preps result in different cell type compositions, so some apparent batch effects between studies are inevitable. The UMAP on the left shows predicted cell types, while the one on the right shows prediction scores. The cells from the reference atlas don't have prediction scores and are shown in gray. This slide gives a brief overview of ChISA, the reference-free LLM-based annotation method that we decided to use in addition to the reference-based method described before. The ChISA workflow starts with clustering the cells, for which we use ScanPy. ChISA annotates the clusters based on their differentially expressed genes. We have started testing ChISA on randomly selected subsets of the Yayon thymus cell atlas, where we can compare its outputs to annotations provided by selects gene. We have been unable to precisely reproduce the clustering that resulted in the original annotations. However, there is a high degree of concordance between the clusters that we get with ScanPy and the original selects gene cell types. We tested ChISA on the high-level clusters and found that its annotations are largely in agreement with the original selects gene cell types. ChISA also has a module that annotates subclusters. We haven't tested it yet. Our next steps are to test ChISA's subcluster annotation functionality, build a semi-automated pipeline wrapping both scVI and ChISA methods, and use that pipeline on a series of datasets relevant to our project. I would like to acknowledge financial support from the National Institutes of Health and from Morgridge Institute for Research. Thank you for listening. Reach out if you have any questions.